Hey guys, I wanted to do a brief overview of the unknown exercise for those of you who missed class that day or who maybe this epidemic started getting to us a little bit before you had a chance to pay attention. So let me just share my screen and I'm going to flip us over to our unknown exercise. This is from your lab manual. This is our unknown exercise. So hopefully you had a chance to take a look at the gram stain. Please note that I've posted all of these ones. Remember that even if you weren't in class that week, for those of you who were absent due to sickness or traveling, if you still don't have an unknown number, please contact me and I will give you one. Otherwise, y'all have your unknown numbers. You can find all of this information you need under unknown exercise. I've posted here all of the unknowns, colony and cellular morphology. So if you click on your particular number, then you should be given access to just a super simple document that gives you the gram stain and what the growth looks like on TSA. So with this information, you can begin working through. I need to move my window here in a second. With this information, you should be able to work through this first part here. Of course, we know that a gram stain tells you all those basic things that we write down at any given time, which is cellular shape and arrangement, and of course the gram distinction. And with this information, you should begin to narrow down the possible list of unknown organisms to just those that match your gram distinction and your shape. I don't have the list with me here. I could provide it if needed for those of you who are absent, but I'm assuming that everyone has already gotten access to the unknown possibilities. So there's 15 different organisms. Your job, first and foremost, is to work through that whole list of unknown organisms and identify which ones are gram positive and which ones are gram negative, first and foremost. And so to find that information, again, going back to our Canvas page, on that same module where I have all this unknown information, I also posted the Burgess lab manual pages here. So all of these first 5, Bacillus, Citrobacter, etc., Enterococcus, Mycobacterium, and Pseudomonas. All of these ones are the actual Burgess pages. For Bacillus, this one will look a little bit different because I pulled this information from 2 different Burgess manuals. This is the systemic bacteriology pages here, and then the next pages, at least for the Bacillus, these are from the systemic bacteriology identification with different characteristics to differentiate the 2 species of Bacillus. But for every other organism, just note that for these Burgess pages, now I've done this for you so you don't have to go and search for where you can find these in Burgess, but essentially I posted for all of these different organisms the introduction pages, which tells you general characteristics behind each organism. So you would start here by looking at Bacillus. And again, the first thing that you're doing, if you haven't started this yet, is work through that whole list of unknown organisms and determine which ones are gram positive and which are gram negative. You could do that just by looking at this first page where you see the genus, and it tells you right here, for example, for Bacillus, cells stain gram positive, and then it tells you some other information too that will be important in developing what we call the biochemical results chart. Okay, going back over to our introduction page. So with this list of unknown possibilities, you're identifying them based on gram positive, gram negative. With your unknown number, you already know whether you are working with a gram positive and gram negative, so you can eliminate out half of those organisms before you do anything else. If you choose to, you can also use the shape of your organism to help narrow down the list of possibilities. And so once you have your group of organisms that you are working with, or your particular unknown possibilities, the next step is to do your homework using these Burgess manual pages. You can use your atlas, you can use your textbook, but I tell you that you can find all the information you need in Burgess manual pages that I put onto Canvas for you. Use these particular tests. These are the ones that we have available for you, or the ones that we have tested the organisms for. So essentially what you'll do is make a chart, right? A biochemical results chart, and it's gonna look something like this. This is in your appendix. You can flip to this if you want to, but I'll just show you what this looks like. So you'll write down the list of your narrowed down list of unknown organisms, either on the Y or the X axis of your table or your row or your column. Identify your organisms with a 1-letter or 1-number simple just to make it easier for downstream when you're doing your dichotomous key. So for example, here are a list of 5 organisms, and the designation given is A through E. And then on the other, whatever, so here is the top titles of each of the columns are the different types of tests that we have available for you, again, based on this list here. So these are the only tests that we have available or have done. Now, I will say that there are a couple of stains that we don't have pictures of up on Canvas. So actually, I wouldn't say on Canvas, on the results that I will provide for you once you submit your dichotomous key and your biochemical results chart. The gram stain you already have, acid fast stain and endospore stain, I don't have those for every organism, but if you need these because they're part of your key, go ahead and email me or text me and I will provide that information for you. So I'll send you a picture of what you would expect to see for the acid fast stain and endospore stain for your organism, if that's on your key. Because those are the only 2 things I don't have for you. Everything else, amylase, urease, catalase, oxidase, all of these different carbohydrates that you're testing for fermentation. MRVP, the SIM test, nitrate, motility, citrate, blood agar, and I think we also have the gelatin. Actually, I have gelatinase, even though it's not on this list. You can add it if you want to, the gelatin test. So all of those ones we've tested these organisms for. I have the results for you. What you're going to do is essentially, again, is to write out, I keep using this word essentially, sorry, to write out all of these different tests or stains up here on the tops of these columns. And now that you have your biochemical results chart listed, and again, it's results for potential unknown organisms, not yours, but all of these ones you're trying to figure out what their characteristics are, so that when you look at the results that I provide for you for your unknown number, essentially you're just going to go and match it up and try to find out which of these things is yours. With this chart made, then you can fill in this information. And to do so, again, you go back to Burgie's pages. Let me give you a better picture, not Bacillus, because this one's a little bit tricky. You'll find, let's say you have gram negative, then you'll click on, there's a couple of different ones that will apply to you. I put most of the gram negatives in the same category here. And so your job is to go through all of these pages and to pull out any information that's relative to what we have available for testing. For example, let's look at Citrobacter here. Citrobacter, this genus, so all of the species within this category, they all fit the same thing. These are all rods, right? There's the size of them, which you don't really need to know. Usually motile by peritrichous flagella. Now this, because it says usually, you actually can't really indicate that as a characteristic. So what you're doing with this introduction is just working through it and trying to pull out anything that's definitive. Facultative anaerobe, that does tell you something. They can respire and they can ferment. So you probably want to think about carbohydrate fermentation. Glucose and other carbohydrates catabolize with the production of acid and gas. That's a really good thing to note. So definitely glucose fermentation with gas production. Here, oxidase negative, catalase positive, methyl red positive. Usually don't regard that one, so you don't want to write this one down yet. Voges-Proskauer negative, lysine decarboxylase, which we don't have. So there's a whole bunch of information right here for you. Reduces nitrates, again, more information. So you can get a lot of stuff directly out of here. But anything that says usually, just put it in the back of your mind, don't write that down. Once you get any information that you can out of this introduction, then you go and find the species information at the table that's listed here. So you can see specific, right here, sorry, specific information about the species on this table, which I've already inputted here. Now, for these pages, I put all of the introductions first, and then I put the table second. So for Citrobacter, once you get that initial information, you can scroll through till you find that table, and here it is. So Citrobacter frendii is the one that we have for you, and this table will give you many of those same characteristics that we just talked about. So oxidase, methyl red, Voges-Proskauer, for example, are here, but there's a bunch of other stuff that you'll find that are pertinent for your chart. Here, anytime you see acid production, that means fermentation, so fermentation positive or negative. So a couple of things to note on this one, anything as you're filling up, taking this information and transferring this over to your own biochemical results chart, you really want to be cautious about anything that says D or anything that has a parentheses or these bars around them. These all mean something a little bit different, but in essence, they all mean that only particular percentage of that species tests positive or tests negative. So because it's not a complete positive and complete negative, I really want you to disregard these ones for the use of identification of your unknown organism. However, you're welcome to still write down the D's and the brackets, etc., for your biochemical results chart. So you'll do this with every organism. Going back over to our appendix, fill up as much information as you can. Note that there's going to be some gaps in your biochemical results chart for sure. As mentioned, you can put in these D's or these little symbols that you see in Burgess, but just note that these symbols, these D's, these types of results means that you really can't use that test to differentiate one organism from another if that is one of your possible unknowns. Let me give you an example of what that means. When you have this biochemical results chart, which is part 1 of your homework assignment before you can get your actual unknown organisms results, this is the first thing that you do. The second thing is, with this information, you're going to build what we call a dichotomous key. And here you're going to take all of your 1-letter or 1-number abbreviation or identifications that you put together for your list of organisms, put it all up here at the top of your key. And what this key is essentially is like a roadmap. If you were doing this in the laboratory, this is exactly how you would go about testing for your unknown organism. So you would start by writing this out on a piece of paper, all of your possible unknowns. And then you go to your chart and find the first test or a decent test that you have all clear positives and negatives for, that can group your list of organisms into kind of a 50-50 if possible. Some of you, it'll be a lot more challenging because of what you're working with, but others, it should be pretty easy. So let's go back up here and we'll just do this together since I don't have you to answer my questions. You can see here already, catalase test would be one good one to start with because you have clear positives and negatives all the way up and down, and you can group these into 3 that are positive and 2 that are negative. The indole test would also be a good one to use, right? A clear positives and negatives and a nice grouping about 50-50, and then citrate would be the other one. Urease is a bad example because we have Borrelia that has no results here, so you cannot use this as a distinguishing characteristic. Here again are 2 others that would not work, MR and PP, because most of these guys are all negative, so you cannot use that as a characteristic definition. And then SIM also would be not able to be used because you have this asterisk mark here. Sorry for the interruptions, guys. So here again are the 3 tests that you could have used. This first one, catalase, citrate, or indole test, because you have those clear positives and negatives. You would do this on your own with your own biochemical results chart. Find that test that you could start off this testing. Write down just one of them. You don't have to do what they've done. You're really just writing down one test. And then you branch it off and show which organisms test positive for that test and which organisms test negative. Here they've chosen catalase, and we can see these 3 organisms, A, B, and E, are catalase positive, so that's what we indicated here. And Vibrio and Borrelia, or C and D, test negative, which would be indicated here on this side. Then you go to 1, as you're working through this, one side or the other. So let's go over here to this catalase negative group, C and D. And for these guys, then you go back up to your chart and say, well, for Vibrio and Borrelia, what is another test that can be used to differentiate between these 2? And you can see there's only 2. That would be the indole test or the sulfur reduction test. Everything else you cannot use because either they test exactly the same or you have urease with no results for Borrelia. So the 2 tests that could have been used here would be the indole test or the sulfur reduction. These guys put motility. We don't even have that on the chart, which is kind of funny, but you would write down whatever it is, and one would be a clear positive, one would be a clear negative, and then you're done with writing out your dichotomous key at this point because you've identified these organisms based on 2 distinct tests. These organisms, if you want to interpret that, would be both of them are catalase negative and one is motility positive, one is motility negative. And if you look back at the chart, only D, for example, is both catalase negative and motility negative. I'm sorry, catalase negative and motility positive. Over here, to finish your dichotomous key, you would go over now to the other side where you wrote down these catalyzed positives, A, B, and E. And now again, you'll have to find a test that differentiates them based on clear positives and negatives that can group them, 2 on one side and 1 on the other. So going back to A, B, and E, looking at your biochemical results chart, can't use catalase, can't use MR, cannot use VP, can't use SIM, right? Wait, you can use SIM. There's indole, A, B, and E. In this case, B is negative and A and E are positive, so that's one you could use indole. Can't use sulfur because you have no results for Acinetobacter, or you have unclear results for this one. You could use citrate. You have 2 positives and a negative, and you could use urease, 2 negatives and a positive. Now, this is a great example of how even though you have a gap in your biochemical results chart, it's still a usable test, because once you've essentially eliminated out D on the other side of your chart, now you can use urease to differentiate between this narrower group of organisms. So in this case, urease was used. We have 1 positive here and 2 negatives. So you're done with E. You don't need to show any more testing for E, but for A and B, you need 1 more differentiating test. And again, going back up here, you could use indole, and that's it. So in this case, there's only 1 test that could be used. And so indole is put here, 1 is positive, 1 is negative, and your biochemical results chart should look like this. There really should only be 1 test at each of these branch marks as you go along the way. And at the end of writing out your key, for me to check it off and provide you with the results, you should have clearly identified each of your potential unknown organisms by a distinguishing test at the end of some type of branching. Some of your keys will look like this, some will be shorter, some might be a lot longer. It just depends on how you put together the results that you see in the biochemical results chart. When you have these guys done, then you're going to submit these, and I have these on our Canvas page. There's an assignment link for the biochemical results chart and the right here, dichotomous key and biochemical chart. So click on this and submit to me both of these things. You can put it all on electronically. You can write it out on a sheet of paper and take a picture or scan it in. I don't care how you do it, just as long as you have both of those things available. Excuse me. As long as you have both of those things completed as best as you can. Now, just note for this unknown exercise, I will only be checking for completion for these 2 things. I do not check it for accuracy. So it's really, really important for you that you get your results correct, and maybe talk and confer with other pod members or something to check your results over before submitting them, or before your actual use of these things in your unknown identification. Okay, let's go back up here. So that's all on page 1. Your homework is to organize them according to Gram-Stain, then recording that stuff, making your biochemical results chart, and making your dichotomous key. Once you have that stuff done and you submit those 2 things, results chart and your key, I will provide for you on your actual assignment feedback. So I'll actually post for you a PDF of all of your unknown numbers results. So it'll give you a picture of all of those different tests that I had just mentioned on the last page. And with that, your job is to work through this information here and record this. And I don't mind if you do this online or if you do it just like we would be doing it in class with your unknown lab exercise notebook entry. But in either case, you're going to be following exactly what is described here. You will have an introduction page with just the basic stuff for lab notebook entries. So put the title, unknown exercise, put your name and the date that you started this thing, the purpose, the methods, and then for your results, this is where you're going to put tons of information. So for your lab notebook entries, there should be, and it says daily entry. Of course, we're not doing it in lab, so it's not going to be exactly the same. But I still want you to do this sequentially, which means do it in order following your dichotomous key. So the first piece of information you should have in your lab notebook entry pages, again, either by hand or on the computer, but it should look the same, are your Gram-Stain and your colony morphology. Those 2 pieces of information you already have available for you. You either need to copy and paste those pictures that I provided for you or draw them out by hand. Either way is fine with me, as long as you include everything that you would normally be doing in your notebook. So that means you need to, for your gram stain, you can't just take the picture and leave it as is. You need to give it a title, and then you need to describe the cellular morphology based on what I ask you to do normally. You can assume that the gram stain images are at 1000x total magnification. For colony morphology, same thing. Draw a picture or copy and paste the picture that I provided for you, but you still need to use descriptive terms to talk about colony morphology there too, right? So that would be the first thing that you have in your notebook entry. The next thing would be moving straight into your dichotomous key, that first test that you have on the top there. So take all of the results that I provide for you in the PDF that I give you. And again, if you're doing electronically, copy and paste the picture associated with the test that you have in your dichotomous key and describe it. So you'll do for each of these things, and then that would be number 5 here. For each of these tests or stains or whatever it is that you have on your key, essentially you're making little tiny notebook entries. So you should, for all of those, have a super brief purpose section. You should have a method section and refer to the actual page in the lab manual where that test was done. So you refer to page blah, blah, blah in the lab manual with any variations that you might have. And then you'll have your results, and that will be the picture that I provide for you, right? So take that image, again, if it's electronic, copy and paste. If it's by hand, just draw that picture out in color, and then state if that's positive or negative, and then do a really brief interpretation as to what that means, right? So my tube turned yellow for glucose. Positive for glucose fermentation means that it can ferment glucose with acid production, something like that. And then do that for each of the tests. So as you're working through your dichotomous key, actually follow the key based on the results that you see, and describe tests only pertinent to where you are on your key. When I'm grading these things, I'm really going to focus on, did you follow your dichotomous key, or did you just throw results up there and describe everything? Because that actually will have you lose points if you're just describing everything I provided for you. All you need to be describing in your lab notebook entry and also in your one-page write-up at the end are the tests that are pertinent to your key, and not just the tests that are on the key, but actually as you follow them from top to bottom to come up with the identification of your unknown organism. So that's number 5 here. So we've talked about all this stuff already. Number 2 is the stuff that you're providing for me on our Canvas page. Number 3, draw the appearance of bacterial colonies. Number 4, draw the gram-stained bacteria and use color, blah, 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 right? So all of the stuff, just follow it to a T, because I literally use this as your grading rubric, and I'll just make sure you've completed everything there. Number 5, we've just discussed this, explain the meaning of your results. Number 6, refer to lab manual and your previous results in conclusion for information about the biochemical test. You're encouraged to discuss any results that seem inconsistent with your conclusions. So if you come to a test, and this might be really important for a few of you guys, some of these organisms in, for example, some of their fermentation are a little bit strange to interpret. If you come to that where one of the tests is really important for you to have the results of some fermentation and you just can't interpret that tube, look back on your biochemical results chart and see if there is another test that can be supplemented for that one first and foremost. If there is, then switch the test. You can ask me and I'll give you permission to do it, but see if you can do that first. And if you really can't, then do the best job you can. Obviously, you have many resources available to you, Google and your photographic atlas, to help interpret those results. But discuss any strange results that you come across. And if you do end up changing your key, the one that you've submitted and I've given you grade for, and if you change it from there, you are required to discuss why you made that change. So if you did come across something exactly like I'm talking about, where an interpretation was difficult to come, or something was difficult to interpret and you decided to switch that test to something else that you had clear results for, that's perfectly fine. You just need to make sure you explain why you did that. And then this last section is the discussion. So for points, if you just look down here, everything here is worth 10 points. So the first one is 10 points, 10 points, 10 points. The final identification means just telling me what organism you have is worth 10 points, and I'll have a Canvas assignment page for just the final ID. So just all you're doing is saying unknown number blank is blank blank, right? Genus and species. And then your last thing that's due is a discussion. And this discussion for us is a one-page write-up, only one page, please. If you're writing it by hand, it's okay, you can do that. It can be a little bit longer, but if it's typewritten, please one page, like 1.5 spacing at minimum, two line spacing is fine, 1.5 pages is what I like. Anyway, so complete this in one page. What you're doing with this discussion are two things. Number one, in your discussion, you're just going to walk me through your dichotomous key in describing your what we call process of deduction. How did you deduce your identification of your organism just following the key? So it's just a way for you to summarize exactly what you just did with your lab exercise. Number two, as you're doing that, I think is the best way to do it, or separately, you're going to interpret your test results into characteristics of the bacteria. So again, for example, if your bacteria was yellow for glucose fermentation tube, then you would say in your conclusion section, in your one-page write-up, I use glucose fermentation to differentiate between this organism and this organism. Mine was yellow, which indicates it is positive for glucose fermentation and produces acid, and I don't know, produces acid from fermentation, I guess you could just say. I mean, that one's kind of an easy one because fermentation is in and of itself what it is. And then you can say, with these results, I was able to eliminate these organisms and focus on these organisms, blah, blah, blah. The next test I did was a citrate test. My organism tested positive, which means it can use citrate as a sole carbon source, and it's a heterotroph. This allowed me to eliminate these ones and blah, blah. So hopefully you see where I'm going with that. So just work through your key, describing the process of deductive reasoning, and then interpreting the results with characteristics of your organism. And all that should take less than a page. Some of you guys are going to get that done really, really quickly, and some will take that full page to write it out. Also in the discussion, make sure that you describe any problems that you encountered. If you got the wrong ID, and actually I will change the date, so you will, just as on the schedule, you'll have your actual ID due 1 day before, or like 2 days before the actual package, the discussion and everything. And that way I can tell you whether you're correct or not. And if you're incorrect, then you can go back and essentially, you don't get points back for the incorrect identification, but you do get points if you can describe your error and discuss what went wrong in your unknown identification. And that would be in your conclusion question. If you also, as mentioned before, if you come across a test in your dichotomous key that has weird results that I provided, and you choose to change that test with something else that has clear results, that's fine, but this is where you would write it up. So I will look for that if you do change it, and I'm looking at your key and I see that it's different from what you uploaded originally, then I will expect to see some explanation in that discussion section. And that's it. So that's 50 points total, 10 points for just the ID, 10 points for the biochemical results chart and dichotomous keys. So those two are sort of separate. The other 30 points will come from the lab notebook entries and then your final one-page write-up. And so these things all have separate due dates for your time frame. I mean, you do have until, I think, sometime in May to submit the final report, but please don't let this get away from you. Jump on top of this. If you can get your key submitted, by the end of the week, next week, by before spring break, then I'll give you your results and you'll have those over spring break to start taking a look at, and maybe even try to tackle this thing before we get into our heavy chapters on immunology, which is coming up next. Okay, if you have more questions, please feel free. All this stuff is just describing what you would be doing in the lab, so don't really worry about that. If you have any questions on any of the stuff, feel free to email me like many of you guys have been doing. Text me if you need to, post questions on Canvas, all that stuff is fine. And good luck with all of this stuff here.